Hi everyone. So we'll start our first session of our initiative R for Bioinformatics. I am Hejat Nani. I am postdoc in Bioinformatics lab at Pasteur Institute of Tunis. We're happy and glad to have with us Dr. Mike Love. So Dr. Mike Love is an assistant professor in the Department of Biostatistics and Genetics at UNC. He earned his doctoral degree in computational biology in 2013 from Max Planck Institute for Molecular Genetics in Berlin. Thank you, Dr. Mike Love, for accepting our invitation and being with us today, presenting an introduction on RNA-seq. So before going any further, we want to introduce you to Our Ladies Tunis. Because Hi everyone. So we'll introduce you to Our Ladies Tunis. I will share my screen. Sorry, I can't see the chat, so I hope it's okay.
Yes, we will take care of the chat. Don't worry, Edia.
Okay. So let's share the screen.
Hello and welcome to this workshop session. Before starting the workshop, we'll introduce you to the Our Ladies Global and Our Ladies Tunis initiative. The R community suffers from an underrepresentation of minority genders in every role and area of participation, whether as leaders, package developers, conference speakers, conference participants, educators, or users. As a diversity initiative, the mission of Our Ladies is to achieve proportionate representation by encouraging, inspiring, and empowering people of genders currently underrepresented in the R community and to facilitate individual and collective progress worldwide. Gabriela de Queiroz founded Our Ladies on October 1, 2012. Our Ladies Global was born and the grant was awarded in September 2016. Since then, Our Ladies has grown to 170 chapters in 44 countries and 39,000 members. Our Ladies Tunis is part of Our Ladies initiative and was created in May 2020 by women working as data scientists and biostatisticians and bioinformaticians. Our goal is to create an R community in Tunis and empower underrepresented genders in the R community. This is the core team of Our Ladies Tunis. Come and join our community on social media. This presentation was made with R Markdown. Thank you for watching this video and enjoy our workshop.
Okay, so thanks again for joining. So I will step back and let Dr. Mike Love introduce you to the RNA-seq.
Great. Thank you so much for inviting me.
Thank you for accepting our invitation. Thank you very much.
I've been looking forward to this. So I have the chat, so I can see if people ask questions while we go. That's fine, and I can see them on my screen down here, and I can try to answer. I want to start by mentioning, like, it's really one thing that's nice in the middle of this very sad pandemic is that I think we've realized that we can have more of these international meetings, like today, very international group. There's a European bioconductor meeting coming up in December, and it's let me put the link here in the chat. So this will be obviously an international meeting. I think the timetable will probably be Europe centric, but you're welcome to look if you're interested in bioinformatics. I really enjoy these bioconductor meetings. There's usually one in US, one in Europe, and there's one in Asia every year.
Yes, actually the Asia bioconductor will be even before, the European one.
Yep.
We can send them the link after because I already registered for it.
Oh, great. Yeah, the Asia one is like, those are more difficult for the US time.
Yeah, that's true, actually.
So I guess, but it maybe for you it's not so bad.
No, it's really bad also.
So just to mention, there's often scholarships for registration. I think the registration fee for Bioconductor in the U.S. was pretty low, but then there's also often there will be scholarships so this doesn't even open until November, but just to mention that. So I have some slides to begin, and let me see. So you can see the slides? Does that work? I guess so. So
Yes.
Yes,
okay,
it works. Yes.
great. And the link for this is down here in the right corner. So it's a bit.ly link. I'll just post this also in case people want to find these slides later. RNA-seq pipe. So I'm going to talk a little bit, take a little bit of time to talk about the pipeline that I'm going to show today for RNA-seq analysis. And then I'll jump into the workflow, which is an R Markdown document. I kind of will jump around in the R Markdown because, you know, we only have an hour today. And I want to make sure I stop and have lots of time for questions. So just one thing from the outset, we probably won't be able to talk too much about the statistical model. That's often difficult in these workshop sessions, is to give sufficient detail on the statistical model. Like when I teach at a couple of courses, and it usually takes a couple of days with multiple sessions to get into all the different statistics that go into these methods on Bioconductor. And so, yeah, I guess if you do want to learn more about the statistical model, you can go all these packages on Bioconductor typically have papers that link out and describe the statistical model. Those sometimes are at a pretty high level, so you may need to like take some intro statistics classes to get into that. But anyway, I recommend you try to read those papers if you're interested in developing your own methods and models. And I guess and then down here at the bottom, I wish I could get rid of this, but I can't. Well, anyway, down here at the bottom, there's two links. There's one to a review paper I wrote I co-authored with a lot of other method developers in Bioconductor. It's called Hitchhiker's Guide to RNA-seq. So there's a long list of people who work on EdgeR and other packages in Bioconductor. And then there's this other link is to Counts model. And that's a chapter I wrote recently kind of summarizing statistical models for count data. So if you did want to read a little bit more about the statistical models, those two links might help. Okay, so so the slides are at this link here. These slides I'm showing are at this bit.ly link that I just posted, the bit.ly./RNA-seq pipe. So I'm going to start by assuming, you know, we have some read data. So we have RNA fragments sequenced from a typically like an Illumina machine. So I'm talking about typically talking about paired end RNA-seq. There are whole new pipelines for long read RNA-seq, but that's not exactly the pipeline I'm showing today. So typically when we have RNA-seq data, and I'm also talking today about bulk RNA-seq data, not from single cells. We have paired reads, which are maybe 100 base pairs long, and we're sequencing both ends of a fragment of RNA. So this is like RNA fragments from a sample, and they're converted into cDNA, and then we sequence both ends. And we have some reference transcripts that we're interested in quantifying. So those are like the human reference transcripts. So those are from the human reference genes. And you can download these reference transcripts from a number of sources like GenCode or Ensemble. Sometimes when people are sequencing rare tissues, so tissues that have not been sequenced before, you might actually discover new genes or new transcripts, I should say. So that's possible, but typically if we're sequencing like a tissue that's been analyzed before, then it's sufficient to look at the reference transcripts. Of course, if you're looking at a different organism, you might also have to generate your own transcripts. So we have the FASTA here is a file format for representing these reference transcripts, And then we have a fast queue, which is how we store these sequenced reads from the sample. And then we'll also need a table that tells us which sample belongs to which group. So treated samples or control samples. And the first step in this pipeline is to run a quantification method called Salmon. And so these are the blue highlights are links. So you can link out to the software package there. This is not a bioconductor package. It's run on the command line. And it takes these fragment sequences and builds you a table where you estimate the assignment of these fragment sequences to different transcripts. So here on the rows, I have, these are the first 3 of thousands of transcripts. And on the columns, these are the first 2 of potentially many samples. And we run each sample through Salmon, and we get these estimated counts. We also estimate what's called the effective length of the transcript, an abundance measure called TPM, and we also do these perform Gibbs sampling to get these things called inferential replicates. That's some technical details maybe. And just the one thing I want to point out about using this tool is that it's both modeling technical biases. So there are various ways in which the read counts could be higher or lower for a given transcript. And those are potentially due to technical biases. That is modeled and corrected by Salmon. And then it also has another method built in called selective alignment, which deals with the fact that some of the reads, so like one thing we've noticed is that genetic variation among samples can lead to differences in quantification. So we found that selective alignment also improves these count tables. So it gets you closer to the true number of counts of fragments from each transcript. So now we have this count table. That's the step where we import the data into R and Bioconductor. So we have a tool called tximport, which will take counts from each sample and import them into an object with a shape like this. So I'm going to talk a little bit more about this in a minute, but we have the counts matrix is stored as something called an assay. That's like a slot in this object. And there's other information attached to this object. So the columns, I said, are samples. So we have, there's a table called call data, which tells us information about the samples. So something here might be, what batch was the sample processed in? Or is the sample a treated sample or a control sample? So just any kind of information we want to store about the samples is attached here. And then we also have information about the features. So those could be genes or transcripts. So those have location in a genome. They might be protein coding or non-coding. So there's different kinds of information we want to have about the features or the rows of this matrix. And then the other thing is, by using this software tximport, we have additional information about these reference transcripts, which we store as a database. So it's kind of linked to a database that tells us information about these reference transcripts. So like what version and where they come from, which is important for reproducibility. So after we've loaded this data into R and Bioconductor, we can do different types of testing. So these are some methods that, these two, DSeq2, I'm going to talk about today. There's another method called Swish, which my lab developed. And then there's a workflow for looking at differential transcript usage. So these are all just different types of questions you might, and methods you might use to interrogate the count data. And for example, if you use DSeq2, you could detect that this is a differentially expressed gene. So actually, we don't need statistics to tell that that's a differentially expressed gene. It's obvious by eye. So the reason why we use a statistical method is because as you go down the list, it becomes more important to know when to say something has evidence of differential expression and when it doesn't. So while we can tell by eye at the top example that it's differentially expressed, that the treated samples have lower counts of fragments than the untreated samples, we want to have a procedure which gives us probabilistic bounds on saying this set of genes has differential expression. And then after we do testing, there are a number of different methods for kind of post-processing the statistical tests. So one example is there's a method from a Wolfgang Huber's group called IHW, which allows you to do multiple test correction, taking into account covariate data. So that's a really interesting method. And I think we have an example of that in the workflow, but we probably won't get to it today. Another method that my group has developed is Bayesian effect size shrinkage. So we post-process the effect sizes and compute posterior effect sizes that are more reliable. So for example, down here is a plot of the effect sizes or the log fold changes of treatment on the y-axis and the mean of the count value on the x-axis. And we've done a Bayesian procedure to shrink the noisy effect size estimates towards 0. So you can see the gray genes that were insignificant in the testing phase. When we perform Bayesian shrinkage, they all shrink towards 0. And we can then, it's much more reliable to use these posterior fold changes if you want to rank the genes. So you're going to say, what is the gene with the highest fold change induced by the treatment? You should use these Bayesian fold changes instead. And then there's another method I mentioned here for different type of multiple testing. So testing groups of genes or groups of features at a time. There's a method called StageR. And then the last thing I mentioned, I won't show that today, but there are a number of different packages for performing, for creating reports. So you can create interactive reports that you can then share with collaborators so they can look up their favorite gene on the fly. And so these are I guess 4 packages here for doing dynamic report generation for sharing the results. And then the one thing I want to point is right here in the middle, before we hand off to these methods, it's actually a nice breaking point, because at that point, once you've imported the data as this thing which is called a summarized experiment, I didn't mention the name of this, you can then take that and use it with many different bioconductor packages. So this is like a breakpoint here, and you can go and use different bioconductor packages, and I'll mention some of those in a couple slides. So this is a code that I use. This is like the code that I use to quantify the reads. So this very first step here, running Salmon, you can run Salmon one sample at a time, or you can use a software called Snakemake to have Snakemake run Salmon for you. And so just to, this might be something that you have to take a look at afterwards, but in green are the input files, in blue are the output files, and then in red are what are called wildcard variables. And here I'm showing you 3 different rules. And Snakemake works a little bit like Make, if you're familiar with Make in Unix. It basically defines a rule that says, if you want to get this output, this is the command that you run, the shell command to run to generate this output. So you define the rules, and then Snakemake, the program, figures out which rules need to run in which order.
Actually, the slide, we are still in the first slide.
Oh, you can't see the...
Yeah, we can't see the second slide yet.
Oh, okay.
Didn't switch to, yeah.
Oh, I see. Yeah, I see. That it popped open a new window. Okay, thanks for telling me.
Yeah.
Can you see this now?
Yes.
Okay, great. So yeah, sorry about that. So green was the input file, blue is the output file, red is these wildcards, and then you can run you run Snakemake, and Snakemake figures out which rules need to be executed in which order. So that's really valuable. And then it also will submit these jobs to a cluster for you. For example, if you have a cluster, and it will, if a job crashes, it will figure out which job it needs to rerun. So I highly recommend, if you end up using Salmon, or I guess any bioinformatics workflow, I really recommend trying out Snakemake. So like one thing, rule all is like, these are the last files that I want to have created. So I want the Salmon quantification files. And then it figures out, okay, to do that, to produce this output, this quant SF file, I know that this rule creates those type of files. So it says, okay, Salmon quant makes that file, but as input, it needs this thing called an index. So how do I create that? And then Snakemake will figure out, oh, that file is the output of this rule. So the first thing I have to do is create the index. So basically, this program kind of figures out the order of operations and runs all these commands. And it won't run a command again if it realizes that it already has that output. Okay, so that, this is a very fast, high-level overview of the pipeline. Let's now assume we've run Salmon. So we've estimated the count of reads for every transcript. And now we want to bring that into Bioconductor. So we've developed a pipeline for that, or a software for that, called tximport. And tximport, so here I just mentioned that we have to quantify the reads. In order to quantify the reads, we first have to create an index. And that index is the reference transcripts, which is a FASTA that you download from some source. So like an example is GenCode. You could go to the GenCode website, download the sequence of the human transcripts, and then index them and quantify them. And you would get a series of files, so Salmon actually outputs these directories. So you get a series of directories of the quantification of every sample. We wrote a software which, when you start to import the data into Bioconductor, it looks at the output of Salmon, and it looks at there's something called a checksum. So checksum is a unique string of numbers and letters, which is associated with the sequence of the reference transcripts. And by looking at that checksum, we can identify the origin of these sequences. And we do that by having what's called a hash table, where we've computed the checksum for many different organisms and versions of reference transcripts. So we realize, we say, okay, we've seen this checksum before. We know that this is human transcripts. We know it's from GenCode, and it's version 29. So that way we know exactly what reference transcripts were used for quantification here. And so we call this function tximport, and it builds what's called a summarized experiment. That's that object I showed with the counts, the column data, and the row data. And it has within it also a checksum that will continually link our data set to the origin of the transcripts. So this source here. And just some notes about how this works. One nice thing is it doesn't matter if the person who did the quantification with Salmon kept track of any details, because it's all stored in that sequence called the checksum. So you don't rely on the upstream bioinformatic analyst to have done any bookkeeping. All you need is the quantification directories. Another thing is that, so this software tximport, it has a cache, which is a database of lots of different metadata files. And if you end up using this software, it makes a lot of sense to share that cache across other users, because then you all benefit from each other's metadata. So no one has to download a file twice because it already exists locally in the cache. And then the last thing I'll mention is this checksum table. Right now we support pretty much all reference transcripts for human, mouse, and fruit fly. But we're also expanding in a way to support many more organisms. If you work with a non-model organism, there's also a solution for you to kind of do this linking of the checksum to the sequence on your own. But I don't know if anybody on the call is also working with a non-model organism. Okay, so I have a little demo which maybe is, I know it will work, so it's better to show the demo. We don't have C. elegans yet, but we hope to within a year, basically. That's a good question. So what happens when you run, let me press pause. So when you run tximeta, it starts importing the data. It found a match and said, okay, your transcripts match with GenCode Homo sapiens release 27. And then it loaded pre-parsed databases from the cache. So after we've run that, we have this thing called a summarized experiment, and it has counts as an assay. It also has information about the rows, and it has information about the columns. So the condition of the sample and the name of the sample is stored in this column data. And it has information about the location of the transcripts, so what chromosome they're from and their range. And something that's nice is that we can very easily, like, if we want to subset to the genes that are on a particular chromosome or in a particular genomic range, we can use very concise syntax, right? So this is saying I want to subset the summarized experiment to only have those genes that are overlapping this range G. Okay, and then the other thing is that because we used tximeta, we, I need to, it's unfortunately, oh, there, I see, get rid of that. So this data set is now linked to the source, the organism, and also to the original files that were used for quantification. Okay. Yeah, so the question about the, so C. elegans or moss, those are not currently supported. It's just human, mouse, and fruit fly that is in this pre-computed hashes. I think within a year, we will be able to link up with the ensemble reference transcripts. So for many different organisms beyond these 3 that we support now, we will be able to compute a hash on our end and match it with a hash that Ensemble creates. But right now, that's the limitation is that Ensemble does not provide. Yeah, we basically need to link up our 2 databases so that we can figure it out on the back end. Okay, so I mentioned there's like a breakpoint after you run tximeta. There's many different tools for analyzing RNA-seq data. And I just put up a number of kind of the most popular ones. EdgeR is another very popular method for modeling count data, including RNA-seq. But there's, and Limavoom, but then there's also these many others. So I just want to mention it's not, DSeq2 is not the only method. There's obviously a lot of other methods in this area. So now I just have a couple lines. So this is like the really simple, how do you run DSeq2? So you load the package with the library command. You turn that summarized experiment into what's called a DSeq data set. So we're just taking that information and kind of putting it into it's actually, a DSeq data set is also a summarized experiment with just one more piece of information. So it has something called a design. The design is a formula. It starts with a tilde, this symbol here, and then it describes how the samples, how the counts might vary across the samples. So the simplest design would be just tilde condition. So if you have 2 groups, you have a variable called condition in the column data. It tells you which group the samples belong to, and you would just say, I'm interested in modeling how the counts vary across condition, so it'd just be tilde condition. In this case, I'm showing a slightly more complicated example where the tissues vary both across batch. There's some kind of nuisance batch effect, and we're interested in controlling for the batch effect and testing for the differences across condition. So in this idea of like controlling for a nuisance variable, we just add it into the design. And so that's actually like how to specify the correct design. We have some information in the vignette, but that's actually ends up being a difficult challenge if you have a very complex experiment. Running the model is one line of code. This DE-seq runs the statistical model, and then we can, from a single run of the model, we can extract different results. So if we want to test treatment over control for the condition variable, we can do that kind of contrast, and we get a table of results called, I named res. And then this posterior shrinkage of effect sizes, we have that separate into a separate function called LFC shrink, which has a little different syntax of how you shrink the coefficient. So this is a really fast, hopefully we'll have time to look also in the workflow. And then I just wanted to show one other example. So this is a different software package, but it maybe has some similar steps. So this is there's a package we wrote called Swish, or it's a method called Swish, which lets you perform statistical testing at the transcript level. So in this diagram, a gene is a region of the genome. So blue, orange, green, and red exons, those form a gene, which is a locus. But a given region of the genome could produce different transcripts. So these are sometimes called isoforms or transcripts. And when we do gene level testing, we're looking at the aggregate output from a locus. So like we're looking at the number of molecules from this locus, regardless of what isoform they come from. But you could also be interested, and you might also be interested in which specific isoform is expressed or differentially expressed across samples. And that becomes a little bit harder because there's a lot more uncertainty in the counts. So basically, when you do transcript level analysis or isoform level analysis, it's very important to be able to take into account the uncertainty of the assignment of these reads across different isoforms. Because the isoforms have shared structure, there's a lot of uncertainty within this gene where those come from. And so we developed a method which has, it's also just a few steps. So we scale the counts. Now this thing contains more information than just the counts. It also contains information about the uncertainty of their assignment. And then we label transcripts that have like a sufficiently high enough count value. This is a method which involves resampling. So you have to set a random seed. And then we run this method, which is called Swish, and you can test against a condition, or you can also control for a covariate. There's even more options in the package. Okay, so let's see. I'll try to finish up, but I do want to mention how to get help. There is function-specific help. If you type question mark and the name of a function, there are vignettes. If you ask for a vignette, it will pop up in your screen, and that's the same thing that you can get online when you click on this HTML link on the Bioconductor website. And then the last thing I have to mention about Bioconductor is we ask that you don't email the developers directly if you have software help, because we get a lot of requests for support. We provide, unlike kind of other platforms, Bioconductor is really good about providing support for users. If they have software questions, you should post it to this website. So there's a website where we answer user questions, and we have kind of a guide for how you can best post a question to Bioconductor. We can't provide infinite support, so we can't, one thing is a lot of people have questions about how they should statistically analyze their data, and there's just not enough time in the day for me to answer all the statistical questions that everyone has. But we do provide software support. So, I got this error, I read the help, I couldn't figure it out, and can you help me out? So more software related questions we answer on the support site. And this is just like the high level important thing about if you have a question, how to post a good question on the support site, or in general, asking questions on any support site. Post your code, that really helps us a lot. Describe your experiment, helps us a lot understand what you're trying to do. And there's this thing called session info in R. And posting that helps us see what version you have. Sometimes people have problems because they're using an out-of-date version of R. Ideally, if you can create a minimal example that you can cut and paste, that's great. Sometimes that's not possible. Okay, the last thing, I have to thank all these people who helped create the pipeline that I'm showing today. And this was really fun slide to make because I realized it's been a really big group of people that work together on this project. So on the left side is people who created the Salmon software. In the middle are people who created the infrastructure for tximport and tximport. Martin Morgan will be your next speaker, so you can ask him about how was it working with Mike. So he's here, he's the director, he's the lead of Bioconductor. On the right side, we have people who worked on DSeq2 and this ape-GLM shrinkage method. And then down at the bottom, we have the team that worked on the Swish method. And then also we get a lot of really great feedback from users. So I want to thank them too. So I'll switch now to RStudio to go over some steps from the workflow. And at this point, I mean, also throughout the talk, but now especially at this point, I'd welcome any questions that people have as we're going. So I'll just start going, but if you do have questions, now is a great time to ask them. So let me.
Okay, so if you have any question, please post your question in the chat.
Great.
I think we have a question. So what do you suggest to use to quantify transcripts for a polyploid organism?
So I guess there's a lot more, my guess would be that there's a lot of shared sequence across the different chromosomes. So like we do have a method, you know, we kind of have a method for everything. So there's a method called Terminus that was just published in bioinformatics, where we say when there's a lot of shared sequence, it becomes nearly impossible to quantify the features separately. So Terminus is a method that in a data-driven way groups the features together at a level that we can resolve them with the data. So it's a data-driven grouping of sequences, and then once you group the sequences, then you can quantify at a group level, and then you can bring that into R and do all the statistics again. So it kind of resolves the uncertainty across highly similar sequences. So while I'll be checking the chat, but I'm also just going to start running through the workflow. So let's see, it's 12:40. The two things I wanted to show in the work were here to kind of run through all the code up until we build this summarized experiment object. Okay, so if I run this line, so just to show you what this is, call data. In this case, it's just 2 samples that I'm importing just for demonstration. We have the treatment, which in this case was dexamethasone, untreated and treated. And then we have other information about the sample. So any information you want to put about the sample goes in this table called call data. And then that's what's provided to tximport, and it will read in the data, try to find a match. If it finds a match, it will download and parse a database, or if it looks in the local cache and finds a local copy of the database, it will just load that. So because there is already a transcript database that I've created beforehand, or tximport created beforehand, it loaded the parsed version. So I guess if someone says that they're confused by long codes, so I guess if you're referring to like these code here, yeah, I guess like if you see a big command, so like if this one is a confusing one, so like here, this one is kind of long. So with basic R, I think the key is to look inside the parentheses and figure out what's being provided to the function. So in this case, we have a function called file path, and then inside there are these 4 pieces. So I think break something down by looking inside the parentheses, understand if you see if you can understand how those arguments are telling the function what to do, and then go from there. So like if for any given line of code, if you're confused, start with the function on the outside, look up the help. So if you want to know, like, what does file path do? Question mark file path. In RStudio, we'll bring up the help, and then you say, okay, so I guess that these things are being provided to ... And so this is, you know, like, I just think that's the best way to get started with, let me see if there's other questions. Oh, no, somebody asked about Snakemake and Nextflow. So I think Nextflow is fine too. So just any workflow manager is a good idea to incorporate into your work. RNA-seq, personal laptop. Yes, the Salmon and Snakemake is good for a laptop level analysis. So Salmon has very low memory costs and memory usage. It takes about 5 minutes per sample. So if you have 12 samples, that's going to take an hour on a laptop. Which method for non-model organism? So we talk about that in the vignette and in the paper for tximport. I'll ask you to refer there. Is it possible to remove batch effect caused by different library preparation? Yes. So again, that would, if you wanted to model a batch effect, you would use a design like tilde batch plus condition. It's using a linear model to control for variation due to batch while testing the differences due to condition. The important thing to think about is that will work if you have a balanced design or not a confounded design. So if all the treated samples are in batch 1 and all the control samples are in batch 2, that's bad. Don't do that. Then you cannot control for the batch effect. You're stuck. So if you approximately balance your conditions within each batch, then you can use these kind of statistical approaches to control for batch effect. Okay, so we made this thing called SE. It's a summarized experiment, and maybe Martin will talk more about this next in 2 weeks. And I just want to mention it has this row information, so it automatically detected and downloaded and parsed the ranges for each gene. That's useful if you want to integrate data with other data sets. Actually have a workflow using Bioconductor kind of link epigenetic data, which would be in the genome, potentially nearby the genes, with the gene expression data by looking at like the distance between regulatory elements in the non-coding genome with the transcription of genes. So just to mention, that's kind of an added benefit of tximport is that it gives you this range data. Okay, and then I'm gonna, so let's see. I think I'm gonna jump down below to where we start working with DSeq2. So so inside of this package the data package I'm using in the workflow, there's a kind of pre-baked data set called GSE. So that's basically, I imported the files and I summarized them to the gene level using that function called summarize to gene. So I'm just loading kind of a pre-baked version of the data, but tximport was used to create this. I'm going to check this function. So
actually, we have 2 questions. So
I see the first one about tximport.
Yeah.
Do I need to read documentation to use it? I think it's important to read the vignette of these software packages before you use them. Especially at the very beginning has usually an introduction about what the software does. But basically, like I've told you today, that tximport is the recommended way to import Salmon quantification into Bioconductor. So, if someone has provided you with files from Salmon, then to go use tximport. is there a function to calculate TPM? So the TPM it's not calculated by DESeq2. That is calculated by Salmon, and that exists already. So here we have this object called GSE, and it has 3 assays, so 3 matrices of data. Counts, which are estimated counts for every gene, abundance, which is TPM, and length, which is the effective gene length. So there's already these 3 assays, and we can look at the TPMs by asking for the assays, and then we can do either $ abundance is one way, or you can also do double square bracket. So this pulls out the abundance assay, which is a matrix, just to show you it's a matrix. So we have 58,000 genes and 8 samples. And just to confirm that this is TPM, you can ask for the column sum, and you can see that they sum to 1 million. So these are, abundance is different than counts. In Bioconductor, we often do the statistical modeling on the count data, but the abundance tells you more about the actual number of molecules in the sample. So let me see. Can I ask if you can suggest a simple tool to detect splice variants, like exon skipping from RNA-seq, if the treatment affected a gene in terms of splicing? So I'm going to ask a follow-up question to Batul. Is this a novel splice variant or part of a reference? Like, is this a known splice variant? Not known? Not novel. So in that case, you could use something like, so in my slides, I had this method called Swish. So Swish can detect isoform level differential expression across conditions. That's just looking like for each isoform of a gene, was there a difference? But then if you follow the link called DTU, that looks to see within a gene, are the isoforms in a gene being used differently across condition. So for example, if when you treat a sample with a chemical, then it's from one isoform to another, that would be detected with a DTU analysis. And so we actually wrote a paper on benchmarking different methods for DTU. We compare for doing that now. So let me see, I'm going to jump below. So we have this, we've imported these 8 samples, and then we summarize the information to the gene level. So now we have counts for every gene and for every sample. We have, in this experiment, there were 4 human donors who donated airway smooth muscle tissue. So it was biopsied. It was created into a cell line, and for each human donor, the cells were either treated with dexamethasone or control. So dexamethasone is it's a glucocorticoid hormone, and it's similar to like what you would, if you have asthma, when you use an glucocorticoid hormone, which suppresses the immune system, among other things. So one thing we would expect in this experiment is that the genes that are related to the immune system might be downregulated after treatment. So that's kind of the goal of doing an analysis. And we have this summarized experiment. It has kind of 2 variables of interest: the donor, that's the human donor for the sample, and the condition, whether it was a little change of the names here. So I changed the donor variable to be called cell. I changed the condition variable to be called dex, just to make, I like to have shorter variable names. And this dollar sign is a way to changing a sample, changing or accessing a sample variable, a variable associated with the 8 samples. And then I also am going to change the names of the levels. So these are kind of long names for the 2 levels of the treatment. So I'm just going to change them to UNTRT and TRT. So you can do this. I can do this if as long as I make sure that I don't change the order, right? So I looked up the order there. That would give you kind of the wrong result. And then I'm going to jump ahead. So in order to make this summarized experiment into a DE-seq data set, we run the DE-seq dataset command, and this is the design. So for today, we want to control for the human donor differences. That's not what we're interested in. We're interested in controlling for that and looking at the difference due to treatment with dexamethasone. So we use tilde cell is the cell line or the human donor identifier, plus dex, and we put the variable of interest at the end. And this, builds a data set. The data set looks very similar to what it did before. Now it just has this extra information about how. So obviously there's a lot of counts. It estimates something called dispersion. That's basically like within a group of samples, how much variability is there for each gene. And it has kind of 3 steps. There are 3 steps to estimating the dispersion. So gene-wise, and then the mean dispersion relationship, and then it does a final estimate that kind of shares information across the genes. And then the last step is it fits the differences in the counts across samples. So it tries to associate the differences in the counts with the variables that are in the design. So because our design is cell plus dex, it's trying to, the last step here is to fit the log fold changes in the counts for every gene dex treatment. And so once this is done, there are a number of different parameters of the model that you might be interested in. So you can ask, there's a function called results names, and it tells you all the different coefficients in the model. So one of the things is it controls for these cell differences, right? So there's 4 human donors. So then there are 3 coefficients that deal with differences between the donors, right? Because if one of the donors is the baseline, there are 3 other donors where the variation might influence the counts. And then this last coefficient is the one that we want to test. So we can call results DDS. If you don't provide any information, it tests the last coefficient. So that builds a results table. And there are a couple things here that are useful. So we have an estimate of the log fold change. That's how much the counts change due to the treatment. And we also have a p-value and an adjusted p-value. And so the kind of the primary output for what most people are interested in is to look at the set of genes that have an adjusted p-value less than some amount. So if we build a results table, we can ask how many have an adjusted p-value less than 0.1. And here, if you run the summary function, it'll print out this table, which tells you the number of genes. And adjusted p-value less than 0.1, that means that we expect 10% of the genes to be a false positive within this reported set. So if you want less than 10% false discoveries, you can lower the threshold here to be 0.01. So there's a question about batch removal in the DE-seq vignette. So this is the workflow here. In the vignette, there's a question in the frequently asked questions section that says, is it possible to remove batches from the transformed data before PCA plot? So we actually have code that tells you how to do that. So if you scan for the FAQ part of the DE-seq2 vignette. I'm gonna pause here, so I'll stop here. Obviously, there's a lot more in the workflow than I am ever able to cover in any given session, but give it a read, and if you have further questions about the software, feel free to post to the support site.
Okay, so thank you very much, Dr. Myklov, for this amazing workshop. Please read the R Markdown, and then if you have any question, yeah, there is a support website of Bioconductor, and you can post there your questions. If you have any other question, now we have 2 minutes of more of questions. So please post your questions now.
And I can actually, let me talk to, I'm gonna ask if I can, I have a meeting with a student next, but I'll just check in with the student and see if it's okay if I stay a little bit longer.
Okay, so thank you very much. Please stay a little bit longer. So yeah, because there are many people, yeah, they are. Thank you. Stay a little bit longer, please.
Yeah, sure. Well, if people have questions, if they have to go, I understand.
Actually, I'm telling them to stay longer because they think thank you, and I
okay,
think
okay.
we're thinking, yeah, to leave the
I'm just
room.
asking if the student, if we start at 1:05.
Okay.
Okay, so there's a question from Carmen. Can you, where you compare one sample against all the others? Let's say you have 5 different tissues and you want to find which ones are upregulated in A. Yeah, so there is a, I think DE-seq will only run if you do have replicates. So if you have replicates for each of the 5 tissues, then you can assess the within tissue variability. So you can then assess if one tissue is, I think that's an important part of the differential expression test. And if you do that, so if you have replicates for each of the 5, you can test if each tissue is different than the rest using like pairwise comparisons, or you can also of the others. And so the way you test, there's a way to test using an average. So if you have results, and then there's you can specify a contrast, which is a numeric contrast. So instead of using names, you can use numbers there, and you can say minus with those. So the -1/4 is computing the average of the A through D, and minus because you want those to be in the denominator. There's a question from PLC, what's the minimum and optimal number of biological replicates? So obviously that depends on the 2 things, the biological variability. So some experiments have very low biological variability, like cell line experiments. Some experiments have a lot of biological variability. So one thing is if you have human samples where you extract tissue like blood from human donors, there's a lot of variability in the abundances there. Like there's differences in the composition of the tissue. So more than just the differences in the expression level, there are differences in the composition. What are the cell types in the sample? So that adds a lot of variability, and so it's important. So like taking that into account when you design power analysis. There are methods, there are some methods in Bioconductor for doing power analysis for RNA-seq. Like I think there's one called RNA-seq power, and then there are more methods. I know that Hao Wu at Emory has a different method for power analysis for RNA-seq. But a kind of a key thing is estimating that variability. And then the other thing is you need to kind of specify, so power depends on the effect size, which you don't know. So you have to guess. So you say, I think that the treatment will induce a fold change of twofold. And therefore, and the biological variability from previous experiments was this value. So there's a way to specify that. Then you can plug that into a formula and it'll tell you you need 5 group to achieve 80% power. And was there another question? You know, the other thing about power, like minimum number of replicates, is there's a paper from it was in the journal RNA, and there they did a really big power tree of 44 replicates, for all biological replicates of a yeast, and they said, when do you achieve, you know, a certain power? And they also profile a lot of different methods, and like, you know, these are the methods that have good false discovery rate and good sensitivity at various, right? 3 samples, 6 samples, 10 samples. So DSeq2, it requires the integer count values. The reason I did that, it's kind of a choice, is because we want people to be putting in counts. If you don't have counts, then the statistical modeling kind of breaks down. So estimate round them because they're just counts with fractional amounts. So we just throw away the fractional counts and we use those as integers. But if you don't have, if you have input data that is not counts in any way, like not fractional counts, it's just something else, you should really be using a different tool, because the distributional assumptions that we make in DSeq2, it makes sense for, you have sequenced a sample and you have counts of those sequences. So, you know, another method that's kind of more flexible is lemma. And so you can use lemma. Someone asked if the output of Callisto is compatible with DSeq2. Yeah, so that's, I'll make a link. There was a paper that we analyzed, well, we maybe didn't run Callisto through DSeq2, but there's a software called tximport, which will import any kind of quantification data, basically, RSEM, Salmon, Callisto, string tie, many different quantifiers. You can then bring it into Bioconductor and use these Bioconductor R packages. Yeah, so I think the important distinction between Salmon and Callisto is that Salmon performs this bias modeling, which we think is really important, accounting for sample-specific biases which modify the counts. We've shown that that's really important and varies across samples and shouldn't be ignored. The other thing is this selective alignment, which helps to deal with the fact that the read mapping is imperfect and some of the reads are best aligned rather than using fast mapping. Any last questions before we cut off? If not,
thank you very much. Thank you for accepting our invitation, and thank you for this amazing lecture. Actually, it was really amazing.
Great.
Yeah, it's actually our first session in the R for Bioinformatics initiative. And we're happy that we got all these participants asking many questions. So it was really a big achievement for us.
Great. I'm very excited to have such an international community of R people and bioinformatics people. So it was great to meet you and hope to chat with you soon again.
Thank you very much. Have a great day. So our next R for Bioinformatics initiative will be on September 30, and we'll have Professor Martin Morgan for our next workshop. So thank you very much for attending our workshop. Stay tuned and please follow us on social media. Thank you very much. If you have any question, you can post your question on the Bioconductor website. So they're there, you know, it's like a support site. You can post your question. Don't post, don't send your mails to the